This video is about the mechanism of urine formation. In other words, the process. How do the kidneys form urine? Well, simplistically, there are 3 steps involved. The first is glomerular filtration. And we've already talked about glomerular filtration previously. It occurs in the glomerulus and the Bowman's capsule. Remember, together they make up the renal corpuscle. And in this step, solutes and fluid flow from the glomerulus into the Bowman's capsule or the glomerular capsule. The second step is called tubular reabsorption, and this actually occurs farther down on the nephron in the proximal convoluted tubule. And in this situation, a large amount of ions and fluids are reabsorbed back into the peritubular capillaries. The last step is called tubular secretion, and this step takes place at the very end of the nephron in the distal convoluted tubule. And in this case, substances are secreted from the peritubular capillaries back into the nephron for secretion as urine. In the next couple of slides, I'm going to go into more detail on these 3 steps, but still keep in mind that this is a very superficial view of urine formation. For more detail, look in your textbook. So again, the first process is called glomerular filtration. This happens in the renal corpuscle, and this is a process in which fluids and small solutes are going to filter from the glomerulus into the Bowman's capsule. Now remember, this is not a particular process. Anything that can fit through the fenestrations in the glomerulus are going to filter into the Bowman's capsule because of the pressure, the high pressure in the blood. The only things that don't filter out are actually too large to pass through the fenestrations, and they include large proteins and blood cells. This next slide is just a kind of a one-page slide that shows all 3 processes. It's really helpful as an overview. So you'll notice that we have here the glomerulus and here the Bowman's capsule. Here is the incoming arteriole, the afferent arteriole, and here is the outgoing arteriole, which is the efferent arteriole. So the first step, glomerular filtration, is summarized very briefly below. And again, in this process, fluids such as water and other solutes such as urea, uric acid, these are waste products, metabolic waste products of most cells, glucose, amino acids, and salts all filter out into the glomerulus. This is the first step called glomerular filtration. Now the next step occurs in the proximal convoluted tubule. Remember, proximal means close to the renal corpuscle, and this is called tubular reabsorption. So briefly, tubular reabsorption is when substances are reabsorbed from the proximal convoluted tubule back into the peritubular capillaries. So from the nephron back into the blood. These are substances the body wants to keep. So again, tubular reabsorption is when substances are reabsorbed, and things that are reabsorbed include sodium, all nutrients such as glucose, positively charged ions, cations, anions, negatively charged such as chlorine. Positively charged would be calcium, potassium, hydrogen ion, and several more, and water. Think about it this way. Anything that your body could reuse in any way is reabsorbed. Your body is a great recycler, and it's going to reuse anything that it can. So of course, calcium you don't want to get rid of, because you can use that for cardiac muscle cell contraction. You also need that for skeletal muscle contraction. Secretion of hormones requires calcium. The release of neurotransmitters requires calcium. Mitosis requires calcium. So you never want to get rid of that. Potassium, hydrogen ions are also necessary. So pretty much all organic nutrients are reabsorbed. In fact, 99% of the substances that were filtered out into the Bowman's capsule are reabsorbed back into the blood at the location of the PCT. Now, reabsorption can be active and passive. However, most substances require active transport. And remember that active transport requires ATP. Passive transport, on the other hand, does not require ATP. And if you remember back to your biology course, different passive transport processes include facilitated diffusion, osmosis, the diffusion of water, and just simple diffusion. So these are some examples. Substances which are not reabsorbed from the PCT back into the blood are chemicals that usually that lack carriers. So in other words, one of the transport processes, facilitated diffusion, requires a carrier to help the substance back. So substances that don't have carriers can't be reabsorbed. If they're not lipid soluble, a lot of times it's harder to get back in, or they might just be too large to pass across the PCT and into the blood vessel. So they might not have been too large to filter out into the Bowman's capsule, but now they're too large to be reabsorbed. Remember that most chemicals, again, are going to require ATP because they require active transport to be reabsorbed. Probably the most important or common non-reabsorbed substances are urea, creatine, and uric acid. Okay, what about the next process? So far we've got filtration, tubular reabsorption, and now tubular secretion. Tubular secretion happens at the end of the nephron in the distal convoluted tubule, or DCT. And what happens here is that substances in the peritubular capillary are secreted back into the nephron at the DCT. So this is the last chance for the blood to get rid of harmful substances or undesirable substances such as urea and uric acid. For example, if your blood is too acidic, this is a chance to get rid of some hydrogen ions if the blood is too acidic. Too many potassium ions, this is a place to get rid of it through tubular reabsorption. Tubular secretion. So let's take a look at the picture again just to see where we are. So again, in the first step, glomerular filtration, solutes and particles filtered out of the glomerulus and into the Bowman's capsule. They then began their process through the tubule, where in the proximal convoluted tubule, they are reabsorbed back into the blood. So 99% of the substances that filtered out are reabsorbed back in the blood. And of course, these are all things that can be used by the body. Then the filtrate continues to pass through the tubule, through the nephron loop, which we'll talk about what that does in a minute, and then through the distal convoluted tubule. And this is where the third process, tubular reabsorption, occurs. So again, last chance for the blood and the body, basically, to get rid of harmful substances, such as hydrogen ions, ammonia, or metabolized drugs. Okay, so those are the three processes. However, we've kind of skipped what happens in this nephron loop. So what does that nephron loop do? Well, the nephron loop, certainly in juxtamedullary nephrons, is involved in controlling the urine concentration and the urine volume. And to explain this, we have to take a look back at the word osmolality. So osmolality is a way of expressing osmotic concentration commonly used in clinical settings. So remember that we said osmolarity is grams of solute per liter of solution. It's a way to describe how concentrated a solution is, but also a way to describe the ability of that solution to attract water. Well, osmolality is a similar term, but it's used in clinical settings. And since many of you are going into the allied health, I'll use this term now. Osmolality is actually the number of particles of a solute per kilogram of solution. So it's very similar. It's, again, a way to measure concentration, but it also reflects the ability of a solution to attract water towards it. Okay? And body fluids, all the body fluids are measured in milliosmole. Of course, milli is 1,000th of a mole. What kind of body fluids are we talking about here? Well, all of them, including any of the fluid outside the cells. Remember, all cells in your body are bathed in fluid. This is called extracellular fluid. Synovial fluid that you find between synovial membranes is another fluid. Of course, cerebral spinal fluid, CSF, the fluid around in the pericardial cavity, your blood. We can go on, but all of this, we're not talking about sweat, by the way, or saliva, but all of these substances are body fluids, and they're all measured in milliosmol. So the job of the kidney is to keep the solute load of all your body fluids at the same osmolality, and that is 300 milliosmol. And this is necessary in order for the different synovial membranes, the serous membranes, to carry out their role. It's also responsible for substances to travel in the correct direction in and around cells. So it's critical that body fluids have this constant osmolality. And the organ responsible for doing that is the kidney. And the way it does it is to regulate the osmolality of the urine. So if there are too many solutes, if the osmolality is too high, for example, the kidney can get rid of solutes and then lower the osmolality within body fluids. Or if body fluids osmolality is too low, the kidney can release less solutes in the urine. So how does it do this? Well, it's a process called the countercurrent mechanism. Let's start by describing countercurrent. Countercurrent is when fluids flow in opposite directions through adjacent channels. So you might think of a river bend. Let's say this is a river. This is a river right here. Here's the land in the middle. The water is flowing in this direction, in this part of the river, and then it's flowing in the opposite direction in the adjacent section. So again, fluids flowing in opposite directions through adjacent channels. This also occurs in the ascending and descending loops of the nephron. Solute concentrations in the nephron loop can range from 300 to 1200 milliosmolar. Now this is only in juxtamedullary nephrons. And this whole mechanism, the countercurrent mechanism, is also only in juxtamedullary nephrons. There are two components to the countercurrent mechanism. The nephron loop is described as the countercurrent multiplier. And this is because as the filtrate flows through the loop, it starts out at an osmolality of 300. It is isoosmotic with all the other fluids in your body. But as it goes down the descending loop, its concentration increases until by the time it gets to the deepest part of the medulla, it is 1200 milliosmoles. So its concentration, its osmolality is multiplying. Now as it goes up the ascending loop, the osmolarity drops from 900 to 600 to 300, even to 100 by the time it leaves. So that's why they call it the countercurrent multiplier. The vasorector, on the other hand, is described as the countercurrent exchanger. And I will discuss this in the next few slides, but briefly they call it this because as the vasorecta comes in, and remember it clings to the nephron loop. It is a straight blood vessel, recta means straight, that clings to the nephron loop. So as it enters the cortex and proceeds towards the medulla, it exchanges water and ions with the area outside of the vasorecta. So if you can think of the vasorecta here, it is exchanging water and salt such that its concentration will mirror the concentration in the medulla, in the medullary area. So we'll explain that in a minute. The other role of the vasorector is the same as all blood vessels. It's to deliver oxygen and nutrients to the tissues in the medulla and remove carbon dioxide and waste. So let's take a look at a juxtamedullary nephron, okay, which is where this countercurrent mechanism occurs. So remember two components, the nephron loop, which is called the countercurrent multiplier. And in this case, you can see the nephron loop right here. So we have the descending limb of the loop in purple, and the ascending limb. And then the vasorecta, of course, is the straight arteriole that basically clings to the loop. And this is called the countercurrent exchanger. Now this particular drawing shows one vasorecta around one nephron loop, but in reality there are many vasorectas surrounding one nephron loop. So there are quite a few blood vessels here. So when we look at this loop, the nephron loop, the cells that line it differ depending on where you are in the loop. In the descending loop of the nephron, the tissues lining it make it relatively impermeable to solids. And what I'm going to focus on particularly is impermeable to sodium. However, the descending loop is permeable to water. Now the ascending loop has different tissue or cells lining it, and so this makes it impermeable to water, but permeable to sodium. And in much of the ascending nephron loop, sodium is actively pumped from the loop into the surrounding tissue. So let's take a look at this all together, the nephron loop here. And you can see in this part here, you can see that it's been divided. Here's the cortex. So everything in this area, everything in here is the cortex. Here's the outer medulla, and then we're into the inner medulla. So again, if you think of this as a renal pyramid, it's a little hard to picture it. Think of it as a renal pyramid like that. And here we're, in this part down here, we're close to the inner part of the medulla. Here's the middle part, and then the cortex out here. So this again is a juxtamedullary nephron. You can see the glomerulus, and you can see that the osmolality is 300. It is isoosmotic with all of the other fluids in your body. Here is the Bowman's capsule, and as glomerular filtration occurs, the fluid or the filtrate in the Bowman's capsule is still 300. It is isoosmotic. As the filtrate moves through the proximal convoluted tubule, remember this is where tubular reabsorption occurs, where solutes and water move from the PCT back into the peritubular capillaries, it is still isoosmotic. It is still 300. But now we're going to enter the descending loop. And as we enter the descending loop, you'll notice that the osmolality inside is becoming higher and higher and higher. It's increasing. And the reason for it is that water is moving out. Now, water is moving out through passive transport, a process called osmosis. And why would the water move out? Well, remember that osmosis is movement of water from a dilute concentrated solution, a dilute solution, to a more concentrated solution. So water is moving down its concentration gradient. So if it is, for example, 400 inside the descending loop, then in the tissue surrounding it, it must be higher. So let's say it's 410. So water always moves from a dilute solution to a more concentrated solution. Okay, that is osmosis. And as the water moves out, the salt is left behind in a smaller volume of water, and so it's more concentrated, higher osmolality. As you move down here, water keeps moving out. Now it's 600. So why would water move out? Well, it must be a little higher. Let's say it's 610. That causes osmosis. So that's kind of what's happening. 900, it must be 910. So as water moves, the inside becomes more and more concentrated. There is more solute and less water, until by the time you get to the inner part, it is 1200. Now, we're going to turn around and start heading up the ascending loop. Remember, the ascending loop is impermeable to water, but permeable to salt. So in this lower part, salt is moving out by passive transport. Well, why is it going to move out? Well, salt is moving by the process of diffusion, which is the movement from a higher to a lower concentration. So there must be lower levels of salt outside, and that's why it moves. Up, as we move up, salt's going to be pumped out through active transport, but again, it's moving from a higher to a lower concentration. Now, as the salt is pumped out, it makes the area outside the nephron loop become salty. And so if you think about it, the ascending loop is producing the gradient in the surrounding tissue. By pumping salt out, you're making the outside 910, 610, 410. So the ascending loop develops the gradient that's in the medulla. It develops it. The descending loop concentrates the filtrate with salt so that the ascending loop can pump that salt out to produce the gradient, which then the descending loop pumps water out so it can produce the salty filtrate. And that salty filtrate is used by the ascending loop to pump out the salt to create the gradient. So it's kind of a circular process here. It's kind of the chicken and the egg, which came first. But this is how the loop occurs. This gradient is really important because what happens as you continue up through the ascending loop is you notice that you drop to 200 and almost to 100 milliosmol. So by the time the filtrate leaves the ascending loop, it is less salty. It has a lower osmolality than it did when the filtrate entered. And that is what the nephron loop does in juxtamedullary nephrons. It produces dilute urine. So the whole purpose here is to go from 300 to 100, and that is the job. Now, once the filtrate leaves the ascending loop, it goes through the proximal convoluted tubule, which remember is where tubular secretion occurs, last chance to get rid of any excess ions or harmful substances, and then it will leave through the collecting duct. So if nothing else happens, the urine leaves at an osmolality of 100. And so again, this nephron loop within juxtamedullary nephrons makes dilute urine. That is its role. Now let's take a look at the Vasa recta, which is called the countercurrent exchanger. Its job is to preserve that gradient that was just produced by the nephron loop. So the gradient that is in the renal pyramid, you want to preserve that, because if you don't, you can't make the dilute urine. And its job is to do that, to maintain the osmotic gradient. Its other role, of course, is deliver nutrients to cells of the medulla via the blood. So how does it work? Well, the Vasa recta, remember, is surrounding the nephron loop. So imagine the nephron loop is in here somewhere, nephron loop. And also keep in mind that outside is a gradient. And the gradient might look something like this. Okay, so we have, let's say that we have, let's start over here, maybe 310. I'm just going to 410, 610, 910, something like that. Now the difference between the Vasa recta and the nephron loop is the Vasa recta is permeable to both water and salt. And it moves very slowly, which is somewhat different than the filtrate. And it also has this countercurrent flow. So what happens is as the blood enters the Vasa recta, it has an osmolality of 300. It is isoosmotic. But then as it starts to get deeper into the renal pyramid, it encounters this gradient. So it is actually saltier outside than it is inside. So since it's permeable to both, sodium chloride will move in because through the process of diffusion, it will go from a higher to lower. And it moves in until the concentration inside and outside is the same, until it is 310 inside. Water, on the other hand, is going to move out. It's going to go from a more dilute solution to a more concentrated via osmosis. And so this will happen until equilibrium is reached, until it's the same inside and outside. And then this will continue down. Now in the next area it might be 410 outside and 400 inside. Sodium is going to move down its concentration gradient through the process of diffusion in. Water is going to move out, attracted towards the more salty medulla. And this will occur until equilibrium is reached. And the osmolarity is the same inside and outside. Okay, in the next part, the same thing is going to happen. Salt is in a higher concentration outside than inside. So it will continue to move in. Water will move out because it's going from a lower to a higher osmolality through the process of osmosis. And this occurs until equilibrium is reached. So this process occurs all the way down until you get to the inner part of the medulla, and then the Vasa recta continues up through the ascending loop. Now the same thing happens. In this case, it might be 950 inside, and it might be 900 outside, or 910. Let's say it's 910. So salt is going to move out because it's going to go from a higher to lower process concentration via diffusion. Water is going to move in to the higher osmolality. That's the process of osmosis, and that continues until the inside and the outside are the same. And it's going to continue all the way back in the reverse, so that by the time the blood leaves the Vasa recta, it's 300 again. It entered at 300, it leaves at 300, and it doesn't remove any of the salt that was within the cells of the medulla. It maintains that gradient. It allows that gradient to stay while delivering oxygen and nutrients, and that's why it's called an exchanger. The reason, again, that the Vasa recta can do this is that, one, it's permeable to water and salt. Two, blood moves very slowly through it so that it is able to reach equilibrium. There is time for this exchange to occur so that you get to equilibrium. And lastly, there's a countercurrent flow. So even though it goes from very dilute to very concentrated, by the time it leaves, it reverses direction. It's countercurrent. It goes from very concentrated to very dilute. And this is how it works. Now, if we look at the entire thing together, you can see again the renal cortex is up here at the top. So here's this whole area is the cortex. Here's the renal medulla. And then here we have the Vasa recta. Okay, Vasa recta. Here we have the Bowman's capsule, proximal convoluted tubule. Remember, the osmolality is still 300. It's isoosmotic. You'll notice that in the tissue between the nephron, it's 300. And then you'll notice again that in the there's a medullary gradient from 300 to 600 to 900 to 1200. So these little particles, let's say they represent sodium, and you can tell just by looking, it's becoming more and more and more concentrated with sodium as it heads down the descending loop, so that by the time you get to the innermost part of the renal pyramid, it is very dilute. On the other hand, as you head up the ascending loop, it gets less and less dilute. So that by the time it leaves, it often is somewhere between 100 and 300 milliosmolar. And now we're entering the collecting duct. Okay, here is the Vasa recta, which remember is exchanging sodium in, water out. Sodium in, here we go, sorry, water out. Water in here, water in here. So again, this is what's called the countercurrent mechanism. A very good review of this is on Mastering A&P. If you look under interactive physiology and then early filtrate processing and just focus on slides 20 to 22, it puts the whole process together for you very nicely. Okay, so after it goes through the juxtamedullary nephron, dilute urine is the default. And remember, the filtrate is diluted in the ascending loop. This is where the dilution occurs. And this dilute urine will then filter all the way out to the renal pelvis. Now, this is what's going to happen as long as a hormone called antidiuretic hormone is not being secreted. Collecting ducts will remain impermeable to water, and no further water reabsorption occurs. So urine osmolality can be as low as 50. That's quite a bit lower than the 300 milliosmolar of all the other fluids in your body. Okay, so the default again for juxtamedullary nephrons is to make dilute urine. But let's think about this. What if you are dehydrated and you want to keep a lot of the water you're releasing in urine? You want to keep that in your blood. Well, in that case, you want to make concentrated urine, which has not as much water and more solutes. If you want to do this, you again have to release antidiuretic hormone. Oops. ADH. And that will inhibit diuresis. Now, in the presence of ADH, by the way, 99% of the water in the filtrate is reabsorbed. In fact, ADH is the signal to produce concentrated urine. So what ADH does is it goes to the collecting ducts and sometimes the the distal convoluted tubule, and inserts substances called aquaporins that allow the water to be reabsorbed. So it inserts those into the plasma membrane, and then water can be reabsorbed from the collecting ducts back into the nephron and essentially back into the blood. So what kinds of things inhibit ADH? Well, alcohol, and many of you might know this. When you drink alcohol, it inhibits antidiuretic hormone, which means that copious amounts of water are released in the urine. And often the next day, people are very thirsty because they're dehydrated. Believe it or not, caffeine can also not necessarily inhibit ADH, but what it inhibits is sodium reabsorption. Back to the blood. Okay, so this isn't really ADH, but caffeine inhibits sodium absorption back to the blood, and so when you have a lot of caffeine, you tend to lose water in the urine. So think of things like coffee and tea and soda. One thing I'll mention here is that in diabetes, there is excess glucose in the blood. So you're losing there's excess glucose in the blood, but eventually that glucose starts showing up in the urine. And when that happens, you have something called glycosuria. Once you have glucose in the urine, water follows, and you start losing a lot of water. That's called polyuria. And then you get really thirsty, and so you drink a lot of water. That is called polydipsia. So people who are diabetic and are not taking care of their diabetes often have excess loss of water in their urine, and it's because glucose is actually showing up in the urine, which draws water with it, and they lose lots of water.